Biology has rules for how cells age, scar, and die. These 9 were not informed. Naked mole rats, Heterocephalus glaber, are almost entirely cancer-free. In over 40 years of laboratory colonies, spontaneous tumor formation has been documented in fewer than 10 individuals, an incidence rate negligible relative to every other rodent studied. They appear to achieve this through an unusually high molecular weight form of hyaluronic acid that prevents cells from overcrowding, a process that precedes cancerous growth. They also survive up to 18 minutes without oxygen, entering a state in which they shift their energy metabolism from glucose to fructose, a pathway used by human fetuses in utero but not available to adult human cells. Their lifespans extend to 37 years, approximately 10 times the expected lifespan of a rodent of equivalent body size. Hydra, genus Hydra, show no measurable rate of senescence under controlled laboratory conditions. In a study by Daniel Martinez published in Experimental Gerontology, 3 cohorts of Hydra were observed over 4 years, and no increase in mortality rate with age was detected across the observation period. Hydra continuously replace their cells through division of interstitial stem cells, maintaining a functionally constant cellular composition. They do not accumulate the molecular damage associated with biological aging in most organisms. Whether this represents true biological immortality or a form of aging too slow to detect within existing experimental timeframes has not been definitively established, but no Hydra has ever been confirmed to die of old age in a laboratory setting. Sea cucumbers, class Holofuroidea, eject a substantial portion of their internal organs through the body wall or through the anus when threatened, a behavior called evisceration. Expelled organs can include the respiratory tree, portions of the digestive tract, and gonads. Predators consume or are entangled by the expelled tissue while the sea cucumber escapes. Organ regrowth is complete within 6 to 8 weeks without any measurable loss of overall function. Some species expel Cuvierian tubules, sticky, toxic threads that expand massively on contact with seawater and entangle attackers, distinct from and in addition to the visceral evisceration response. Multiple evisceration and regrowth cycles have been documented in the same individual over a lifespan. Planarian flatworms, order Tricladidae, can be cut into over 200 pieces and each piece will grow into a complete, functional organism within 2 weeks. The regenerated individuals are not blank copies. Research by Shamrat and Levin published in Journal of Experimental Biology demonstrated that planarians trained to navigate a spatial environment, then decapitated, with the head regenerating from the body stump, relearned the trained task approximately twice as fast as untrained controls. The implication is that information from the original nervous system influenced the formation of the regenerating brain. The mechanism by which a headless body stump retains and transmits learned behavioral patterns to a newly grown brain has not been explained. Spiny mice, Acomys species, are the only confirmed mammals that can regrow tissue through regeneration rather than scarring. When full-thickness holes are punched through their ear pinnae in experiments modeling tissue loss, the holes close completely within 30 days, with regeneration of skin, hair follicles, cartilage, and blood vessels, indistinguishable from the surrounding original tissue under histological examination. Standard mammalian wound healing produces a scar from fibroblast activity. In spiny mice, fibroblast recruitment is suppressed and replaced with a regenerative program observed otherwise only in amphibians. The genetic pathways responsible have been partially identified and are present but silenced in other rodents, suggesting that mammalian scarring is a derived trait that evolved secondarily from an ancestral regenerative capacity. Bdelloid rotifers, class Bdelloidea, have reproduced entirely without sex for an estimated 80 million years, an evolutionary duration over which no males have been documented in any of the approximately 450 known species. All individuals are female and reproduce by parthenogenesis. They avoid the genetic stagnation expected from long-term asexual reproduction through an unusual mechanism, horizontal gene transfer. Up to 10% of their active genome consists of DNA acquired from bacteria, fungi, algae, and plants, integrated into functioning chromosomes and expressed as normal cellular proteins. The organisms accomplish this integration by desiccating almost completely when dry conditions occur, partially fragmenting their own DNA in the process, then rehydrating and repairing the fragmented DNA, incorporating environmental DNA in the same repair cycle. Antarctic icefish, family Channicthiidae, are the only vertebrates known to lack hemoglobin as adults. Their blood is transparent. Oxygen is carried dissolved directly in the plasma, a mechanism with much lower oxygen carrying capacity than hemoglobin-based transport, partially compensated by a blood volume approximately 4 times higher than an equivalent-sized fish, an enlarged heart, and capillary vessels expanded to 3 to 4 times the typical diameter. At the near-zero temperatures of Antarctic water, dissolved oxygen concentration is higher than in warmer seas, making the hemoglobin-free system viable in this environment and nowhere else. They also lack myoglobin in most muscle tissue, making their flesh translucent. Their hearts are pale. Approximately 50% of their total metabolic cost is cardiac, keeping the oversized heart running. The Pompeii worm, Alvinella pompeiana, lives attached to the outer walls of hydrothermal vent chimneys at the bottom of the Pacific Ocean, in an environment measured at 80 degrees Celsius at the worm's tail end, while the head, in the ambient water column, is exposed to approximately 22 degrees Celsius. This is the highest confirmed sustained temperature tolerance of any complex animal. They are not heat-resistant in a passive sense. The worm actively maintains this temperature gradient across its own body length. The mechanism of thermal tolerance, specifically, how proteins in the posterior end function at 80 degrees Celsius without denaturing, has been studied for decades and remains incompletely understood. The bacteria living in a mat on their backs may play a role in thermal management, but the specific mechanism has not been established. Axolotls, Ambystoma mexicanum, can regenerate functional cardiac muscle following injury. Experiments in which portions of the ventricular wall were surgically removed showed complete regrowth of the excised muscle, restoration of normal cardiac function, and integration of the new tissue into the existing electrical conduction system within 60 days. They can also regenerate portions of the brain, including sections of the telencephalon, and the regenerated neural tissue integrates functionally with the existing circuit. In both cardiac and neural regeneration, the axolotl does not first form scar tissue and then replace it. It suppresses the scar formation pathway entirely and substitutes direct regeneration. The signaling molecules that activate this pathway are present in mammals, including humans, where they are expressed during fetal development and then shut down permanently at birth. 9 animals. 9 sets of rules that biology apparently forgot to enforce. Tomorrow's animal does something that should be physically impossible. This channel posts a new animal fact video every day. Subscribe so you don't miss the next one.